All right, so grade 8, now that you've learned about cells and the parts of cells, we're actually going to talk about how things move into cells, out of cells, and in cells. So basically, like, how do things get around the cell itself? How does it get the nutrients it needs, the waste, and so on? So just going to the next slide, we're going to talk about what you hopefully should learn throughout this. By the end of this topic, and this is going to be a 2-day topic, but you should know the differences between permeable, non-permeable, and selectively or semi-permeable membranes. You should know how diffusion works. Okay, so you're going to learn about diffusion. We're going to actually do a little experiment, and you're going to learn how that works. What can affect diffusion? So what can we actually do to change the rate of diffusion, how fast or how slow it happens? And then later on, we're going to get into how osmosis works, how osmosis is similar to diffusion, and what affects osmosis. And then finally, we're going to talk about how this applies to plants and talk about things like transpiration and how stuff gets into the roots and so on. So it's a big topic. We're going to be dealing with this again for a couple of days here, but it's a good one. All right, so individual cells carry out the same activities as whole organisms. Cells make use of the water you drink, the air you breathe, and the food you eat in order to carry out their functions. So just as you need to take in things and get things out, cells need to do that as well. Materials passing into the cell are checked, and that's in quotation marks because it's not actually an active process that it gets checked, but different properties of that material will determine if it gets through or not. So essentially the cell membrane controls, not actively, but it controls what gets in and what gets out of the cell. And this all happens because of the structure of the cell membrane, the makeup of the cell membrane. It contains this very special molecule that's called a phospholipid that you don't need to know the specifics of, but basically this phospholipid actually allows the membrane to somewhat control or regulate what goes in and what goes out, and then stop other things from going in and go out. So let's talk about the terms permeable, impermeable, or selectively or semi-permeable. So impermeable, when I say that something is impermeable or a membrane is impermeable to something, I'm saying that that substance can't get through. Okay, so an umbrella is impermeable to water. Okay, water cannot get through the umbrella membrane. If I say that something is permeable, that means that the material can get through it. So if I say that, looking at the picture, that shirt is permeable to water, that means that water can get through the shirt, right? It can travel through the shirt so the person feels wet through the shirt. So that would be permeable. It means that something can get through. And then finally, if I say selectively permeable or semi-permeable, then that is a membrane allows some things to get through and other things not to get through. So if a membrane is selectively permeable, that means it stops some things and it allows other things. So if I look at the Canada-US border, this is essentially like a membrane, right? The border will stop some things from getting through and it will allow other things to get through. So for example, it might stop drugs from getting through or firearms from getting through or other stuff, but it's permeable to other things. Like for example, air, right? Air can get through just fine. Citizens can get through, although that's a longer process going through the actual border guard process, and so on. So some things can get through just fine, while other things cannot get through, or they have to go through a process to get through, which is a great analogy, analog to the cell membrane, where some things can just get through easily. Stuff like water can get through the cell membrane easily. Gases like carbon dioxide, oxygen can get through easily. And then some things are checked or can't get through at all. So things that might be checked are things like proteins or carbohydrates, viruses, right? That kind of thing is checked, and then from there, either it will be able to get through or it won't be able to get through, and the membrane will be impermeable to that thing. So again, selectively or semi-permeable means some things can get through while other things cannot. So what is the cell membrane responsible for? Well, we learned that it is for keeping things in or out of the cell. It's for keeping the inside of the cell different in composition than the outside of the cell. So it creates these separate environments in and outside the cell, and it does that by regulating what goes in and what goes out, by trying to control the in and out of the cell. So just like kind of a bouncer, a cell membrane is selectively permeable. It only allows certain things like water, nutrients, and gases to go in or out of the cell, and it will stop other things from going in or out. So it will be permeable to the stuff that the cell needs to be permeable to in order for nutrients to get in and waste to get out. And it will be impermeable to stuff that it needs to be impermeable to. So all of the inside of the cell doesn't leak out and harmful things don't get into the cell. So it's both permeable and impermeable, so it's selectively permeable. I thought it would be interesting for you to actually see a diagram of a cell membrane. You don't have to know much about this, and in fact, you learn a ton about the cell membrane in Science 10. But what we have here is something called a phospholipid bilayer. So this right here, one of these is a phospholipid, and that phospholipid has a phosphate group and then lipid kind of chains down here, and it has what we call a hydrophobic tail, which is this, which means it doesn't like water, and then a hydrophilic head, which is this part, which does like water. But that doesn't matter. Just kind of neat that the actual chemical composition of this, the fact that it's made of these phospholipids and these channel proteins and stuff, actually allows it to do what it does. So just neat. It's all about the chemistry. So we're going to talk about diffusion, and then we're going to get into actually a little mini kind of lab thing around this as well that hopefully will work out well and you guys will participate in. Sorry, just bear with me as I try to get my mouse to work. We're going to skip this. But diffusion is directly related to the particle model of matter. We talked about the particle model of matter in chemistry. This really applies to this as well because it's based on the fact that particles are constantly moving around. And you hopefully remember that point from the particle theory of matter, and that as temperature increases, particles are moving more. So particles are always in motion. They bump into each other more often when they are close together until they spread out evenly. Movement from an area of high concentration to an area of low concentration is called diffusion. So if I have something that is more concentrated in one area than another area, it's going to start spreading out through collisions of particles until it's even everywhere. This diagram does a really good job of showing this, and we're going to talk about concentration in a second. But really what concentration is just the amount of stuff I have per unit of area. So let's say that this was a bus, okay, and let's say that everybody's crowded towards the front of the bus and the back of the bus is just empty. Well, eventually what's going to happen is that people are going to end up spreading out until they're evenly distributed throughout the bus. Just like people, particles want to get elbow room. They want their personal space. So because of the collisions that occur and the movement of particles, they're going to end up evenly distributed roughly as time goes on. And this just naturally happens. It doesn't require energy from the cell or anything. Just things move from where they're more concentrated to where they're less concentrated over time. So let's talk about concentration. What concentration is it's how much of a substance is mixed with another substance. So it's how much there is of something per unit of volume. And I have this red square here to kind of show you the difference. If I treat this red square as a unit of volume, right, this area in the red square is quite concentrated. That would be a high concentration because I have lots of the solute, the little dots, per unit of solution or volume. But if I move this red square over here, well, this is now a low concentration. I don't have much here in terms of dots, solute, per unit of solution. I have less. So this is higher concentration, and this is lower concentration. So what's going to happen over time? Well, over time, these particles are going to move around by colliding and moving until the concentration throughout this is equal. Okay, so there's the same amount everywhere of solute per unit of solution. An analogy for this, let's say you make microwave popcorn, right? So you make it, and at first it smells the most in the kitchen where your microwave is. But as time goes on, the whole house smells like microwave popcorn, and that's because the particles of the microwave popcorn have now distributed evenly throughout the whole house. And then it starts escaping to the outside. And over time, more and more of those particles of the microwave popcorn make it outside, right? From where it's high concentration in the house to where it's low concentration out of the house. Until as it goes out to where there's less concentration, eventually your house doesn't smell like microwave popcorn anymore because it's all diffused out. I remember when Axe body spray was just such a huge thing in schools where mainly actually like boys in junior high, grade 8, grade 9 would just go like all over themselves with Axe body spray and the whole hallway would reek. That's a great example of diffusion. It went from where it's more concentrated, right, on the kid after spraying himself all over to where it's less concentrated all throughout the rest of the hallway in the school. Right, so particles move to where there's more to where there's less. And actually talking about smelling, when you smell something, that's because particles are actually going into your nose. You're actually getting the thing in your nose, which makes farts incredibly more disgusting. When you smell someone's fart, you're actually getting their fecal matter in your nose. Yay! All right, so how do cells use diffusion? Let's say we're talking about something like an amoeba, and this kind of blob is an amoeba. We're going to pretend. Normally there is a balance of carbon dioxide inside and outside the amoeba, just like this. So we could say that generally the concentration outside and inside is pretty much the same. I've got the same amount of dots per area, inside as outside. Maybe it's a little bit higher inside, but basically the same. Well, let's say that the amoeba makes a ton of carbon dioxide. Well, all of a sudden now there's a lot of CO2, a lot of carbon dioxide inside and not very much outside. So what's going to happen? Well, what's going to happen is that over time, this is going to end up moving out. It's going to diffuse out. So these particles and their collisions and their movement are going to end up going from where there's a high concentration to where there's a low concentration. And that's diffusion. That's all it is. It's just things moving from where there's a lot of it per unit of volume to where there's less of it. So diffusion will occur until the particles are evenly spaced out in a container or across a membrane. Some other examples. Laughing gas is released at the front of the class. Funny, right? Because both laughing and yeah. You fart in a car. Don't do that. Your friend pees in a pool. These are disgusting examples. This is horrible. But if your friend peed in the pool, over time, the pee would be evenly distributed throughout the whole pool. Hooray. And hopefully it's chlorinated. Also get new friends. All right, so we're going to talk about a lab. I want to actually try this out. This is going to be interesting. We're going to do kind of like a little bit of a hybrid between like doing this and then I want you to try something out in person. Little mini lab here. And I don't love that I actually have all the answers here because I wanted to talk about it and I wanted you to come up with it. So don't look. I know you can rewind and pause, but pretend like you can't. All right, so the question or the problem we're dealing with here is how does temperature affect diffusion? How can we actually change the rate, how fast diffusion happens with temperature? Does it change diffusion? So we're going to start out, and I want you to put this in your notes. Can you please rate a hypothesis? What do you think will happen? So how will temperature impact diffusion? So what I'm looking for here is a statement of, will it go up? Will it increase the rate of diffusion? How fast diffusion happens as temperature goes up? Will it go down as temperature goes up, or will it stay the same? Okay, now we're going to talk about variables. Variables are something that you may not have talked about much before, but this is so incredibly important in science and the scientific process for us to understand variables. And I think I actually changed the order of this, so just bear with me while I change this slide just slightly to match. So we're going to come up with a manipulated, a responding, and two controlled variables. So what is a manipulated variable? A manipulated variable is what we are changing in the experiment. So we're trying to test the effect of something. In this case, it would be temperature. So what we're manipulating in this experiment is we're manipulating the temperature of the water. And we're going to use water here. So we're going to manipulate the temperature of the water. And we're trying to see what the impact of this temperature change is. So what do you think the responding variable is? What do you think it will be? And the answer is the responding variable is going to be how fast diffusion occurs. So I skipped ahead a bit here. We're gonna test this out somehow. And I think the best way for us to test this at home would be for us to actually have some hot water and some cold water. And then we can use something like food coloring or just something that has a very strong color that we can put in there. Maybe you have flavored drink crystals or something that has a color, whatever. Gatorade powder. As long as it has a strong color, we can make this work. So what we're going to do is we're going to put some food coloring or something colored into hot water and cold water, and we're going to see how fast or how long it takes for the color throughout that glass to look the same. So if I have a glass of hot water and a glass of cold water, what are some things that I have to make sure are the same between what I do with the hot water and the cold water in order for it not to mess up my experiment? So what are some things that I need to control? And that's what a controlled variable is. It's what are some things I need to keep the same between the two glasses of water in order for us to have valid results. And one thing that we definitely need to control is the amount of water. We need to make sure the amount of water in both glasses is the same. Another thing that we need to control is the amount of solute. We need to make sure that we put the same amount of food coloring or solute in both glasses, right? So both glasses. So those would be two things that are incredibly important. I would also say that the shape of the glass is important. If I had a very, very tall, skinny glass compared to a thick kind of tumbler glass, it might end up giving different results, right? As it kind of spreads out and diffuses. So the shape of the glass would be something that we want to control as well between the two trials. Okay, so I think you should go try out this experiment, take a look, and I want you to record how long it takes for the food coloring. So take a glass of hot water, glass of cold water. Make sure it's the same amount of water. Put food coloring in both glasses at close to the same time. And then watch and see how long it takes for both glasses to get to the point where we can say it's uniform in both. So in other words, the color looks the same throughout all of the water in that glass. Give that a try. And that's it for today. That's diffusion. Hopefully you got quite a bit out of this and understand what diffusion is. It's really just things moving from where there's a higher concentration, more per unit of volume, to a lower concentration. And hopefully you also learned about permeable, impermeable, and selectively permeable membranes, and you know what those terms mean. All right, that's it for today. Have a good one.